So let's get started. We had to, sorry for the change in the schedule. I actually think it's not so bad. We'll break up the biology lectures a little bit. Just to review where we are in the overall program. Yeah. Oh, thanks. Okay. Okay. How's that? Can you hear me now? Is it good?
Okay, yeah, you're good.
Okay. So we've talked about the causal model that underlies everything that we're going to be talking about. We've done some of the biology that explains why the assumptions of the model are good approximations. And then David talked about drawing inferences about the effects of the genetic factor as a whole. And now what we're going to do is switch to, so that was developed in the era before there was access to genetic data itself. Now we're going to begin to talk about the actual analysis of genetic data. This lecture and the next one that I'm going to give are about approaches to gene discovery, which is about how to identify the links between specific genetic variants and phenotypes that we're going to be interested in. I'm talking about it not because we think that's something that you're going to go out and do, but because it's important to understand how we learn about those links, since you're going to presumably be doing applications that build on the knowledge that we have from gene discovery. So it's important to be able to evaluate the work that you're going to be relying on and know which is more or less credible work. And it'll also give you a bit of historical perspective on the field. I think for a social scientist, genome-wide association studies are is an unintuitive approach to doing empirical work. And I think the kind of background we're going to talk about in this lecture will help put that in context and make it clearer why that's the preferred approach. Okay. So actually, the very first thing I want to do is say something about genetic data, since we're going to be talking about using that kind of data. From our point of view as users of genetic data, it's actually pretty simple. You collect a tissue sample. It used to be blood was the kind of tissue you'd need to collect, and then you needed a specialist who knew how to draw blood. Today, what's much more common is saliva samples, which are very easy to collect. You can just get someone to spit into a test tube, and then the saliva preserves the DNA well for several days at room temperature, so you can just mail it to a lab. And then when it gets to the lab, the DNA is extracted, it's copied many times, and then a machine reads the genotype data from the DNA. So this is something that Andrea is going to talk about in more detail later today. The common approach today for the data we're going to be talking about is genotyping. It costs about $30 a person. And with genotyping or genome-wide genotyping data, you're measuring a set of genetic variants that are pre-specified on an array, and they're chosen in a way to capture most of the common variation across people. These days, they're typically measuring about 2.5 million SNPs, and then you can impute that data up to about 10 million SNPs, which I think is something David's going to be talking about in a few days. And then the other technology that's worth knowing about is sequencing, because it's getting cheaper and cheaper. That's where you measure, either literally measure every base pair in the genome. That's how we often think about sequencing, like with the Human Genome Project. But what's likely to be even more common in the very near future is what's called low-density sequencing, where you don't sequence every base pair, but you take a random sample of base pairs, and you sequence those. And so that is as cheap as genome-wide genotyping, and you actually get more than 2.5 million SNPs by doing low-density sequencing. Yeah?
In a world where we're only concerned about SNPs, what would be the advantage of like a low-density sequence versus just building a bigger array with 10 million probes?
So the question was, what's the advantage of low-density sequencing over building a bigger array? And there's two main advantages. One is that the genotyping arrays are biased to capturing common variation in European ancestry samples. And because you take a random sample with sequencing data, you no longer have that bias. The other issue is that when you get a large enough sample of the sequencing data, you can build you can estimate well the LD matrix of the entire genome, and then you can impute from the low density sequencing data up to a much higher density, essentially like getting sequencing data as if it were high density sequencing data. Okay. So today it's mostly genome-wide genotyping data. And then there's 4 kinds of data sets that are worth knowing about that tend to have this kind of data. The first is cohorts, which is the term that's used in medical genetics for data sets, that are focused on studying medical genetics. So they're interested in cancer genetics or cardiovascular disease genetics. And then similarly, twin registries, which were around before genotyping, but many of them have genotyped their participants. And they're typically in the size range of 500 to 5,000 or so individuals. If you want to work with this, so when we first started doing GWAS, this was all of the data that was available that was genotyped. And so you'd have to collaborate with these cohorts if you wanted to analyze genetic data. Now, as I'll mention, there are other sorts of data sets. If you want to work with this kind of data, you generally have to collaborate with the cohort. They have strict privacy requirements, and often they even have to do the analysis themselves. So here are some examples are the Framingham Heart Study, the Swedish Twin Registry. Second kind of data set are social science surveys. So this is in the last 5 years or so, 5 to 7 years. Some large social science surveys have started to genotype their participants. Health and Retirement Study was an early adopter. The Ad Health sample that you're going to be analyzing, also relatively early in getting this kind of data. Today, there's also the Wisconsin Longitudinal Study, English Longitudinal Study of Aging. There's actually a number of social science data sets like this. These data, typically these data sets are larger, about 5,000 to 20,000 people. Sometimes they're representative samples. You can often get direct access to these data by applying, although there's stricter IRB and privacy restrictions than for accessing the general phenotype data. Third kind of data set are national biobanks, and these have really only come on the scene in the last couple of years, 5 years ago, because the UK Biobank was the first and became available in 2016. But now there's a number of others, the Estonian Biobank, the China Kadoorie Biobank, and a lot of other countries have initiatives to create biobanks, including the US. These can be much larger. In the case of the UK Biobank, it's 500,000 people. That's also the aspiration of some of these other biobanks. In the US, the goal is to get to 1 million people in the All of Us project. These are also data sets that you can usually get access to through some kind of application procedure and maybe paying a fee for processing the application. And then the fourth kind of data set are personal genomics companies. I should have updated this N. I will in the slides that I fix on Dropbox. The sample sizes now can be much bigger than 500,000, several million people in some cases. So these are companies like 23andMe, Decode Genetics, MyHeritage. So this is where they're providing a service to customers, like giving you your health status or your ancestry information. And in return, a number of customers, well, they pay, but then also many of them consent to participate in research. And then the companies analyze the data themselves, and in some cases, collaborate with researchers to analyze the data. For a number of these companies, there's application processes that you can go through to potentially get access to this data. Yeah.
About, I guess just a couple slides ago, just about genotyping.
Yeah.
You said it's $17 or $30 per person, but how can we do research on universities that genotype other people's data, or that we can outsource it to?
So your question is, how do you actually
research what companies could genotype our data?
What companies could genotype your data? There are there are some companies like 23andMe you can just get directly, although it's fairly expensive. They'll do the genotyping for you. GenCove is a company that does low density sequencing, and they'll do it at around $30 a person. Most of the time when researchers do their own genome-wide genotyping, they work with a lab that's associated with the university. Many universities have labs that will handle the genotyping for you and provide you with the equipment. Yeah, so if you're interested in doing it, actually, I can put you in touch with some people who will be able to direct you to the best resources for these days for doing your own genotyping. Okay. All right. So any other questions on the data or the data sets before we turn to the main topic of gene discovery? Yeah.
Is there any effort to harmonize the variables in the social survey? I mean, like, I was thinking about, like, let's say we want to use like, polygenic scores for peer education assignment, but it may be the case different social surveys ask the education question in a different way, like whether you went to the college or simply asking years of education, etc. And I was just wondering, is there any necessity to harmonize the data, or if so, like, is there any effort to do that?
Yeah, so the question was about harmonization of the data. For some of the data sets, for the data sets related to aging, there's an initiative called the Gateway to Global Aging that harmonizes the phenotype data. That's freely accessible online. That doesn't cover all the data sets that you potentially be interested in for the genetic data. So in some cases, most cases, you just have to do it yourself. For the polygenic indexes, we have the SSGAC has an initiative called the Polygenic Index Repository that harmonizes the way that the PGIs are constructed across a number of different data sets that you would potentially use. Yeah.
One question. So for the social science data sets, in most cases, it's like a subsample of the data set that have like all this data. Is there like any correlation with like age or like, I mean, because I'm guessing that especially for HRS, it's kind of like a new thing. So if there's like how much of it is like a random sample or also social space data sets?
So are you asking which ones are representative samples?
Yeah, no, but I mean, like all these like social science surveys have like their own survey design and all that, and then like a subsample of that is the one that gets that. And I don't know like to what extent it's like uniform that people are like like agreeing to like.
I get the selection in
Yeah.
being genotyped within a sample.
Oh, selection being genotyped. Yeah. So there's definitely some, I mean, you know, almost surely there's some selection into who agrees to be genotyped. In some cases, the HRS had a particularly high response agreement rate to be genotyped. I think it was around 75% of people who were asked agreed. In most of the other data sets, the social science data sets, I believe the consent fraction was lower. I don't think it's, you know, I haven't seen that selection well characterized in any particular way, and you know, I mean, I guess you could reweight the sample depending on what you're doing to try to get a more representative sample type result.
I do remember Jessica Paul saying once that like some of the individuals were like so old that they couldn't produce enough saliva to get the DNA. I don't know how big of a problem that was, but when they were first collecting, I remember she said something like that.
Okay. So Patrick was just saying that there's some in the HRS, apparently some people were too old to produce enough saliva. Okay. Okay, so gene discovery. So we've talked about heritability estimates, but they don't tell us which genetic variants matter, or how much they matter, or why they matter. They don't allow us to construct the polygenic indexes that we can use in other data sets. So gene discovery is the process of identifying associations between genetic variants in a phenotype, and it's a necessary step before we can apply genetics in social science research. Okay, so we're going to focus on estimating the additive model. And there are two reasons, a kind of cost reason and a benefit reason. So let me start with the cost of trying to extend beyond the additive model. So the problem is that if we, if with the additive model, we have one parameter to estimate for each SNP j. If we try to estimate dominance effects, then we have two parameters for each SNP. And if we want to estimate epistasis, then we potentially have an enormous number of parameters. I mean, even just if we want to do additive by additive epistasis, you have, it's an explosion of possible parameters, and that's just, one simple form of epistasis. Okay, so the problem with more parameters is we have to test more hypotheses. The main problem is we have to test more hypotheses, and as we're going to talk about later, the correction for multiple hypothesis testing is extremely important in gene discovery. And if we needed to correct for every time for twice as many hypotheses, for example, to look at GWAS, it's going to really adversely affect power. Now that may be less of an issue for dominance going forward as sample sizes get larger. It's still a major problem for epistasis. So the other issue is on the benefit side, and that's I think we alluded to this earlier. There are a number of arguments, reasons to think that the additive model is going to do pretty well in terms of capturing much of the variance, at least for many complex phenotypes. There's a number of reasons for that, and in the reading list, there's a paper by Goddard, Ray, and Vischer, I believe, that goes through systematically all of the arguments. I want to focus on what I think is the most important, probably the most important argument. And for this, I'm going to switch to a slide from, Patrick, could you switch to David's slide from his heritability lecture? It's slide number 18. So this is something I think David mentioned, but I want to highlight it more. So this is a slide that David showed when he was talking about what the additive model is and how it's the best linear predictor. And what these three figures are showing is how the best linear predictor changes as you change the frequency of the trait increasing allele from 50% in this figure to 75% to 95%. So what happens as you go from the left to the right is that more and more of the data ends up in this genotype, where you have 2 of the trait increasing allele, and less on the 0 trait increasing allele. So the best linear predictor line shifts its slope. It gets flatter and flatter. And essentially, when you get to 95%, it's just trying to fit these 2 points, and it's essentially ignoring this point here.
Yep, when I share it, it interrupts your sharing.
OK. Oh, I didn't. OK.
I barely got to where your slide.
OK. Well, this is fine. So if you look at how well you're fitting, how much of the variance you're explaining, in this case, since there's essentially no data here, you're getting a very high R squared from this regression. And you can essentially perfectly fit, as this frequency of the allele tends toward 100%, your fit is going to get asymptote toward perfect because you're just fitting 2 points with a line. And of course, you can always fit 2 points with a line perfectly. So in the genome, of course, depending on the SNP, the frequency of the trait increasing allele is going to vary. You could think of it as a first approximation as uniformly distributed, so uniformly distributed between 50% and 100%. But that means there's going to be a lot of SNPs like this, where you're capturing almost all of the variance with a linear model. So what this means is that even if there are dominance deviations in much of the genome, the additive model is going to capture most of the variance, including capturing variance that's due to dominance effects. The same issue, the same kind of argument can be made with epistatic effects. It's more complicated, but it's the same basic idea that if you have 2 different SNPs and the trait increasing allele for both of those SNPs is high, the frequency of the allele is high, then ignoring the interaction is going to be fine. You're going to capture most of the variation that you would get from having the interaction. Okay, so there's reasons to think that the additive model is going to be a good approximation for complex phenotypes, and it's costly to try to estimate the additional parameters. So that's why the focus has been on the additive model. Should I can unshare my screen. Is that what I'm going to?
Oh, it's not about you sharing or unsharing. It's just going to get, once I start sharing, I'm going to.
Oh, I see. OK, that's fine.
So just give me 1 second. OK. So you should be able to. This work.
You share?
Yeah. So you should be able to share, and I think people will see.
OK. OK, so we're going to focus on the additive model and try to estimate these average effects, these beta J's. That means we're going to potentially miss variance that matter via interactions. If the interactions average to 0, we might also miss variance that matter but have maybe they're over-dominant effects, and so they average out to 0. Yeah.
Yeah, so you mentioned that it's computationally intensive, and also you lose a lot of power when you probe these epistatic effects. Is there any value in taking top SNP hits and the ones that we really see an effect for, and then kind of building a model around that where you test interactions with some other genes and maybe take a more targeted approach at looking at epistasis, or is that kind of a weird candidate gene way of looking at things?
No, so the question was, what about focusing on the top hits that you get from an additive model and then seeing if they have dominance effects or epistatic effects? I think that's a perfectly reasonable strategy. You know, there's no, so I guess the kind of implicit theory underlying that is that there are some variants that matter for the trait, and then a lot of ones that don't. So let's find the ones that matter, and then the ones that matter might have these more complicated kinds of interactions. So I think that's perfectly sensible. You know, you still could miss a lot that way. I think Patrick was telling me that this was actually done in the recent, which paper was it that did this?
It was done in the Ripke paper on
In
schizophrenia many years ago, yeah.
In a 2014 schizophrenia paper, they took that approach to try to look for epistasis, and I guess they found very little, right?
If you plot was just flat.
Yeah.
Cool, thanks.
Okay. So there are three major problems that come up when trying to do gene discovery. The first that we've already mentioned is multiple hypothesis testing. And there's a few different reasons for this being a problem. One is that we often measure many genetic variants in a data set. We're going to test up to 10 million SNPs in a GWAS, for example. Sometimes there's several different phenotypes that you could analyze. And for any given phenotype instead of SNPs, there's lots of different specifications that you could adopt or control variables that you could include and not include. The second problem, we used to say, and I realize I said this on an earlier slide, population stratification, but I want to be more general here and talk about gene-environment correlation. Population stratification is one case where gene-environment correlation comes up, but there's also potential indirect effects from parents. There was that example that we had from the early exchange with Tammy about parents potentially having alleles that lead to less nurturing behavior that might lead to aggressive behavior in the children. That would be a kind of gene-environment correlation that would confound identifying a causal effect of the SNP. So the question is whether we have control variables that are sufficient to eliminate all of those kinds of potential confounds from gene-environment correlation. And then third is low power. So do we have a large enough sample given the true effect sizes, which I'm going to argue tend to be very small, and the significance threshold, which to deal with multiple hypothesis testing is going to have to be very stringent, and I'll get to this toward the end of the talk today, a prior probability of any given SNP having a strong association that in most cases is pretty low. Do we have a large enough sample to have adequate power? Okay, so in the social sciences, there have been two main approaches. In social science genetics, there have been two main approaches to gene discovery. One is the candidate gene study, and the other is GWAS. So I'm going to get to GWAS in the next lecture. Today, I want to spend a little bit of time on candidate gene studies to understand where we as a field went wrong. And this is something that Paige talked a little bit about in her talk, some of the appeal of the candidate gene studies in the early days. Okay, so a candidate gene study is where you specify ex ante hypotheses about a small set of SNPs. In many cases, it was just a single SNP based on its believed biological function. So Paige mentioned a number of reasons why candidate gene studies might have been politically attractive to researchers. There are also scientific reasons they were attractive. First of all, this sounds like regular science. You know, you specify a hypothesis and you test it. Sounds like good. It is good science often. It also used to be very expensive to measure genetic variation. In the days before GWAS, the days before SNP arrays, you couldn't measure a large number of SNPs. And even in those early days, it was very expensive to measure many SNPs. So measuring a single variant was much less expensive. OK, so you estimate a model where you include just that single SNP in the model. You set a significance threshold of 0.05, sometimes corrected for multiple hypothesis testing, maybe the number of SNPs that you test. And until recently, this was virtually all existing work in social science genetics. It's still a surprise. There's still a surprising number of candidate gene papers published every year. But here's a timeline of how social science genetics compared to medical genetics in terms of the approaches to gene discovery. So basically, before 1990, there was relatively little gene discovery work going on because it was so expensive to measure any genetic variant. In the 1990s, there was a growing amount of work in medical genetics using linkage studies, and then they were often followed up with candidate gene studies. So let me just give you one example of how that worked in the case of Alzheimer's disease and ApoE. So for late onset Alzheimer's disease, there were linkage studies that had been done that identified a particular region of the genome that seemed to be associated with ApoE. So remember linkage studies where you have data on families and you follow the disease through the family. Late onset Alzheimer's disease is not Mendelian, but it turns out that there is this variant ApoE that has a big effect. So you could see it, you could detect it in these linkage studies, even with a relatively small number of families. And then there were some researchers who were thinking about, what could be the gene in that region of the genome that was leading to Alzheimer's disease. And it was known that Alzheimer's disease was characterized by apolipoprotein plaques that build up in the brain. And one component of those plaques is beta amyloid proteins. And thinking about what was known about the genome there, you could see that there was a sequence of base pairs that coded for the beta amyloid protein. And so that led researchers to hypothesize that that region of the genome, the gene ApoE, may be what's responsible for what was being detected in the linkage studies. So then there were candidate gene studies that followed up and confirmed that ApoE was, in fact, the responsible gene. So when I was first getting involved in social science genetics, which was around 2000 or so, when I was starting grad school, that was the shining example that was for social science genetics. We want to find these genes of big effects and use a candidate gene approach to help us identify, confirm good hypotheses for which genes are mattering. The first GWAS was done in 2002, at least the first high-profile GWAS. There was a major GWAS that was done in 2005 that was very successful in identifying some of the biology underlying age-related macular degeneration, which is where you lose eyesight in older age. Then there was a landmark study in 2007 that set a lot of the modern methods for doing GWAS. And by this time, the linkage and candidate gene studies were becoming increasingly rare in medical genetics. In social science genetics, there were basically never any linkage studies, and I think part of the reason for that is that you don't have these case-controlled designs that you use in the linkage studies, so it's not clear what you follow through the family tree for the kinds of phenotypes we're interested in. So the focus was always on candidate genes. The kinds of hypotheses that people had were not, they were things like, well, the dopamine-related gene, we know that dopamine is involved in decision-making and the reward system in the brain, so it's reasonable to think it will be related to risk-taking behavior. So that's or novelty seeking. And so that was the kind of hypothesis that underlay the candidate gene studies in social science genetics. And you can see, that the first GWAS was the GWAS of educational attainment, the EA1, published in 2013. So there was a long period of about 10 years when medical genetics was increasingly moving toward GWAS or had fully switched to GWAS, where social science geneticists were still doing candidate gene studies. Okay. So the ApoE example shows that the candidate genes approach, in principle, makes a lot of sense. But in medical genetics, actually, in this period, in the early GWAS period, the linkage and candidate gene studies were getting less and less replicable. And what was happening was that the earlier medical genetics had been focused on Mendelian diseases first, and then things like ApoE, where there were genes they were not Mendelian, but they had these genes of large effect that contributed. And then they were trying to study things like depression and schizophrenia, which were, turns out, complex phenotypes where there are no genes with big effect. And the linkage studies were underpowered, and so they produced bad hypotheses for the candidate gene studies, and those results tended to not replicate. And in social science, the replication record was never that good for the candidate gene studies. So when I got involved early on, candidate gene studies were the state of the art. I did a candidate gene study in data from Iceland. We put together a chip that measured about 300 different candidate genes, which were all dopamine-related, serotonin-related, or brain-related genes, and measured them in a sample of about 2,000 people. And then I was a graduate student. I went to Iceland. I coded up all these social science phenotypes from this was a medical cohort, because that was the data that had the data sets that had the genetic data, and looked for associations between all of the 20 phenotypes that I'd coded up and the 300 candidate genes that we had measured, and found something. And we were very excited. It was actually an association with educational attainment. So we started this work around 2004. We've finally had this association around 2007. We had some collaborators who were medical geneticists, and they said, well, in our research world, there's been some bad experiences with candidate gene studies, and we don't really believe them anymore unless you replicate. And so we then undertook a replication in 3 other data sets, including an additional 2,000 people from the same Icelandic sample, and nothing. The result didn't replicate. So then, my own evolution in thinking was, my collaborators and I wanted to understand how widespread this issue was of non-replication. It was not widely understood at the time that there were any problems. Led by Chris Chabris, who's a psychologist, David is also a co-author in this paper, a number of us set out to systematically look at associations with candidate genes for cognitive performance. So we looked through the literature. There was actually a literature review on all of the candidate gene associations with cognitive performance. So we identified all of the genes that had been associated. And then we looked at 3 data sets we had access to that collectively had 10,000 people, which was big at the time. Most of these candidate gene associations were in data sets of about 100 people. So we had much more power than the original studies to try to replicate these associations. There were 12 genetic variants that we had in our 3 data sets. And so here's what we found. What I'm showing you here on the x-axis is the 12 different SNPs that we used as markers for the candidate genes. The y-axis is the association size with cognitive performance pooled across the 3 data sets with the 10,000 people. And you see the effect estimates on units of IQ points per minor allele with 95% confidence intervals. And you can see the 95% confidence intervals all overlap 0. So that's to say we didn't find evidence for any of these candidate gene associations. Moreover, these effect sizes, we're ruling out non-tiny effect sizes. The standard deviation of IQ is 15. And typically, the candidate gene associations that have been reported were pretty substantial in magnitude, standard deviation or more. OK. So, yeah.
On the previous slide, have any of these SNPs or anything in LD with these SNPs, like in the current era, are we finding any significant associations now with these things, and it was just a question of power, or we were just totally off?
So Grant is asking whether any of these associations have shown up in the GWAS era. I actually don't know for these particular ones. I wouldn't be surprised if there are some true small associations. But I think the associations are likely small enough that effectively these were false positives in the original finding, because the effect size was close to 0.
Right.
We did actually ask your question in the risk GWAS that we ran. We looked at the candidate genes that had been associated with risk and looked at how strongly they associated with risk tolerance, and we could, they were not.
Okay.
They were essentially not associated at all. I think there was some growing recognition around this time, although this was actually pretty early in 2012 that this happened. But the editor of a leading field journal, Behavior Genetics, understood that there was a problem with the candidate gene studies at the time, and so it adopted a new policy for this journal that required replication of any candidate gene studies that were going to be published, and also put in some standards for how they had to control for population stratification and address multiple hypothesis testing. But let me just read to you a couple of sentences of the introduction to this new policy, because it's very strongly worded for an academic editorial policy. He wrote, "The literature on candidate gene associations is full of reports that have not stood up to rigorous replication. This is the case both for straightforward main effects and for candidate gene by environment interactions." And he cites a paper by Duncan and Keller, which was a non-replication of a lot of psychiatric genetic results. As a result, the psychiatric and behavior genetics literature has become confusing, and it now seems likely that many of the published findings of the last decade are wrong or misleading and have not contributed to real advances in knowledge. So this was a first recognition by the community that we had gone down a wrong path. A number of other journals quickly followed and adopted the same policy as Behavior Genetics, including Psychological Science, which is a leading psychology journal. Despite this, it actually, candidate genes remained the main method for doing research in social science genetics for a number of years. I think partly because there was no real alternative at the time. Okay, so what was the problem? Well, I think the fundamental problem was with the premise of a candidate gene study, which is that you're going to have a few genes with large effects that you're going to be able to detect in the sample sizes that people were using, where I'm saying it's about 100 to 3,000. 3,000 is really on the extreme large side of the candidate gene studies that were done at the time. What we now know is that the effects of any particular variant are very small. And it seemed, one thing that added to the credibility of the candidate gene studies were these hypotheses that seemed reasonable. But the problem is when in retrospect, certainly, when you think about it, the idea that a dopamine-related gene should be related to risk-taking is a pretty weak hypothesis, given that there's all sorts of other genes that are involved in brains and how the brain operates that you might also have hypothesized were going to be involved. So if you add up, if you think not just about that single hypothesis in isolation, but the whole set of hypotheses that you could have formulated, then the strength, the effect size you should expect from that particular pathway would have to be pretty small. Yeah.
So this might just be a confusion, but so in your Icelandic sample, so the way we think about social traits, so there may be, like there may be many different pathways to achieve higher education attainment. Like it might depend on different social settings, but even in the same social setting, two people can achieve like the same, say, GPA, having very different kinds of skills, or be more risk-taking through different types of motivators. So I'm wondering, like in your Iceland example, what if actually the genes that you identified for that particular group, they were probably geographically recent, maybe or maybe not, maybe they were spread all over the Iceland. But, so what if those were actually the correct variants, but in other people, other variants matter, and what we're doing with GWAS is essentially picking up all of those and just creating an average score. So, so you and I may have the same average score, but I'm getting that score because of variant A, and you're getting it because of variant B.
Yeah. So the question is about heterogeneous effects, and maybe the reason why these results don't replicate is that in one population there is an effect that we find, and in the second population there isn't an effect because because there's different mechanisms for the associations. So that's always a possibility, and we can't 100% rule it out. But I guess I would argue two things. One is that we're always getting average effects, and what we found, whether we're doing Kennedy gene studies with GWAS, and what we found with the GWAS results is that they replicate quite well when you have enough power in your new sample. So it's not clear why that should be a property of GWAS associations and not Kennedy gene associations.
So what I'm saying is that because the way the GWAS score is calculated is just assigning weights. So say if there are 70 SNPs that are identified, and I have, say, 60 of those, and you have a different, say, 55 of those, our SNPs don't overlap, but we can achieve the same risk score for whatever phenotype, right? So it will replicate in GWAS because we can achieve the
So I agree that you could find that it's likely the polygenic index is going to have better replicability than the individual SNPs. Yes, but GWAS are not identifying polygenic indexes. When I talk about the replicability of GWAS, I mean the specific variants that are identified in the GWAS have replicated well. The other issue I would say is that we now know that this relative to the sample sizes, relative to the true SNP effect sizes, the sample sizes are so small that were done in these studies that the confidence intervals are huge relative to the distance between true genetic effects and zero. So, I mean, I think, I'd rather not say, I think more correct than saying that these were false positives, I would just say these were uninformative data. Just such large confidence intervals. Yeah, maybe there's an association, maybe there isn't, but we basically didn't learn anything from a study with a sample size of 100. Yeah?
I was wondering, whether the problem, especially in the early days of cognitive approach, especially in the social science traits, is to do with the assumption of small genes having larger effect. Wasn't this known already that these are complex traits and
Yeah, so the question is, wasn't it known that these were complex traits and that genes should have a small effect? So it's interesting, we probably should have known, but at the time, all of the success stories in genetics were cases of finding these genes of large effect. And everyone looked to those as what it was going to look like to discover associations with other phenotypes. So people expected the future to look like the past. And even when people imagined complex traits, there was a time in the 2000s when people were saying the advantages of GWAS for complex phenotypes. They were saying, what if these phenotypes are polygenic? Like there's 10 variants that affect these. That's what people had in mind as polygenic. Maybe we should have known, because in retrospect, Fisher in 1918 was imagining a continuous distribution where every variant literally has an effect on the genome, so it's millions of them. But that was not what people had in their heads in the mid-2000s. Okay, so the candidate gene studies were not addressing these three problems. The multiple hypothesis testing was actually, it seems like it was pretty common. What people would do is look for a different candidate genes for a given phenotype and then only publish the one that came out significant, or they would look at, different, it wasn't necessarily a single researcher doing this, or it wasn't necessarily intentional. It could be different researchers came to these data sets because there were only a handful of data sets that had these candidate gene measures. People would test different hypotheses, and some of the groups would just give up their projects and go home, and other groups that found something, they would be the ones who published their finding, and this was uncorrected for. This is, of course, more generally a problem in the social sciences, as we're seeing with the current replication crisis. Gene-environment correlation, it was understood that this could be a problem, but it was very difficult to do anything about it. You couldn't create principal components, which is a way we're going to talk about that GWAS tried to deal with this, or use family data. There wasn't much of that. Typically, the best you could do was restrict to self-reported whites or control for ethnicity. But that's for reasons we've talked about and we'll talk more about. That's generally not going to be sufficient. And finally, low power, which I think is really a really central issue. And so I want to go into more detail on that because I think understanding power is important very broadly for the things we're going to talk about for the rest of the course. Okay, so I want to start with a power analysis building on, which you can all do, having done the problem set with power calculations. Let's imagine that we're going to test a candidate gene with a significance threshold of 0.05. Let's imagine the true effect size is an R squared of 0.02% of the variance. That is about the largest effects we're seeing for SNP associations with educational attainment in large GWAS. And let's imagine we do a big candidate gene study, so 3,000 people in our study. So what is our statistical power to detect this association? Well, it turns out it's 12%. Okay, so abysmally low by standards for what is reasonable power. The sample size you would need for 80% power is 40,000 people, much larger than any candidate gene studies that were done. Okay, so why is that a problem to have low power? Well, first I want to just mention some common misconceptions that people have about power that you often hear mentioned by researchers. Well, first of all, remember what power is the probability that you find a statistically significant result given that there really is an effect, and you calculate it using the true effect size. Now, when you estimate power, you need to use, you don't know the true effect size, so you need to come up with some plausible effect size ex ante. One thing that you often see people do is they run a study, estimate an effect, and then they take their estimated effect and use that to conduct a power calculation to say, how well powered was I to have found this effect? And the problem with doing this is that if you found a statistically significant result that's just by chance, you're now using a large effect estimate to calculate what your power would have been, and you're going to way overestimate what your power was. So you should never use an ex post estimate of the effect size to calculate power. What you need to do is look at existing evidence in related contexts to make a best guess. There's a nice paper by Gelman and Carlin that I'm going to reference on a upcoming slide that has an example of how this can be done. So one misconception that you often hear is that power matters for whether or not I'm going to find an effect when there really is one. But once I found an effect, I don't need to worry about power. So we're going to talk in a moment about why that's a misconception. There's an even more extreme version of this, which is that if I find an effect despite having had low power, then my result's even more convincing. It's kind of like an overcoming adversity sort of idea. It's totally backwards. Okay, so here's the problem with low power fundamentally, and that's what I drew on the whiteboard to illustrate the point I'm going to make here. So low power is equivalent to saying that the true standard error of the estimate, of your estimate, is large relative to the truth. The truth. So there's some true beta, it's not 0, and low power means that the uncertainty around your estimate, this what I'm showing here is a confidence interval, is large relative to the distance between the effect and 0. But this is high power, this is low power. So if the standard error of your estimate is small, this high power situation, then it's very likely that your estimate is going to end up being close to the truth. And it's likely that the confidence interval around your estimate is going to exclude 0. Okay, so that shows the fact that it's likely the confidence interval is going to exclude 0 is exactly saying that you have high power. Okay, so that's the equivalence between small confidence interval, small standard error, a small true standard error, and high power. Okay. Now, if, on the other hand, the standard error is large, then your estimate is going to be uninformative, not telling you much about the true value. And in that case, you can't conclude much from the fact that you didn't reject 0. So that's what we mean by low power usually. It's like the fact that we didn't reject 0, we wouldn't have expected to reject 0 even if there were an effect. But it also means that you can't conclude much if you did reject 0, because there was a high chance you were just going to end up in the with some big effect size. And that's what people often don't understand about low power. If you think about it this way, you can see low power, you're just getting an uninformative estimate, whether it's significant or not. Okay, so I want to mention 2 manifestations of this problem. One is what Gelman and Carlin call sign and magnitude effects, sign and magnitude errors. So they talk about 2 kinds of errors that you can have with a statistically significant result. One is a sign error. They call it a type S error. That's the probability that you estimate the wrong sign of the effect. And then the other kind of error they talk about is a magnitude error, or expected type M error. And they talk about that in terms of what they call the exaggeration ratio. So that's the ratio of the magnitude of the effect that you estimate relative to the truth. So if that's bigger than 1, it means you're, regardless of the sign, you're expecting to exaggerate how different, beta is from 0. You're expecting to exaggerate the magnitude of the effect. So they show that when power is lower, both of these error types are larger. And we're going to talk more, Patrick's going to talk about the winner's curse. It's very closely related to the exaggeration ratio. So the idea is low power. When you find a statistically significant result and you have low power, it's likely that you've overestimated the effect. Yeah?
I have a question on maybe if we think of like a single snip versus like a haplotype. So in terms of like power, we're thinking about like a single snip, right? Whether we are hard enough to identify like a significant snip. But sometimes like some SNPs are like inherited together. So whether combining like different SNPs would maybe allow us to be more powered? Is there a way or any kind of work that's
Yeah. So the,
like haplotypes instead of just the synthesis.
yes. So question is, can you increase power by studying haplotypes instead of SNPs? So there are a number of methods for doing that. They haven't been adopted very widely in GWAS studies. I mean, I think I'm actually not entirely sure why. Does anyone know why?
I mean, GWAS kind of is doing that a little bit in that when we do GWAS, we're capturing everything that's an LD with the SNP that we're looking at. If you wanted to,
Yeah. There are those methods like I forget what it's called, that tries to measure the gene, the gene level tests instead of.
tests?
What's that?
Like burden tests? Or?
No, not burden tests, but there's gene level tests that you can do. You kind of weight the SNPs that are involved in the same gene or something. I mean, I guess that's not exactly the same as a haplotype. Yeah, so it may be just what Patrick said, that you're getting pretty far along in that direction anyway with GWAS. Okay. Okay, so Gelman and Carlin have some calculations in their paper of how the likelihood of these different kinds of errors depending on power. I'll just highlight with the kind of calculation we did before the counter gene study with 10% power, there's something like a 5 to 10% chance of just getting the sign wrong, and you expect exaggeration of something like 2 or 3 times the effect. So the second manifestation is a high risk of false positives. So power is about the probability of finding a significant result when there really is an effect. But what we really care about is the probability that there really is an effect, given that we have a significant result. So is the hit a true finding? And so we're going to talk about how you calculate the likelihood of a false positive. To do that, we need to switch from thinking about power to thinking about what the truth is. And in order to switch the order of probabilities, we need to use Bayes' rule. So here's a reminder about Bayes' rule, and there'll be a problem set. There's a problem set question on this, I think, in problem set 2. So for any 2 events A and B, the probability of A given B is this ratio, the probability of A and B divided by the probability of B. And then we can substitute in for the probability of B, the probability of B and A, and the probability of B and not A, the sum of those 2 things. And then we can use, again, this definition of conditional probability and get this formula for Bayes' rule. Okay, so we're going to assume for the calculation, we're going to try to make the things as simple as possible to get some intuition. We're going to assume there's 2 states of the world. There's either a true effect, and we know the effect size, which is some beta, or there's no effect. Now, if you're going to do these calculations in practice, Bayesian calculations in practice, you'd want to specify a distribution of effect sizes. But again, this is we're simplifying here. So the information that all we're going to have is whether we found a significant effect or not. Now, in practice, I think Patrick's going to talk about some of these things of essentially how credible effect estimates are. In practice, you'd want to take into account what the actual estimate is, but we're going to imagine all we're looking at is whether it's significant or not. Okay. Okay, so the probability then, using Bayes' rule, the probability that we have that there really is an effect given that there's a significant result, this is just using the formula from before, it's the probability that you have a significant result given that there really is an effect times the probability that there's a significant effect, the prior probability, and then the same thing in the denominator plus the probability that there was a significant effect given that the null hypothesis is true, that there's no effect, times the probability that there was really no effect. So we can write that, we can divide through by this term. And then that gives us a probability of H1 given significant. We can go through the same exercise to get the probability of H0 given significant, and then divide one by the other, and that gives us an easier formula to remember, which is that the probability of there really being an effect given significant divided by the probability of no effect given significant, which you could think of as the posterior ratio. So this is the relative likelihood that there really is an effect relative to no effect, given that we have a significant finding. It's the product of two terms. One is the probability that you would have, the relative likelihood of getting a significant result under when there really is an effect relative to no effect, times the ratio of the prior probabilities of an effect to no effect. This term, probability of a significant result given that there really is an effect, is power. And this term, the probability that you get a significant result given that there's no effect, is the significance threshold. So this ratio of power to the significance threshold turns out to be an important quantity. It's called the rejection ratio, and it can be interpreted as the strength of evidence from rejecting the null hypothesis. So in particular, it's the strength of evidence in the sense that you start with some prior. It's this rejection ratio that when you get a significant result, the rejection ratio is by how much you should now increase your weight on there really being an effect relative to no effect when you're updating to your posterior ratio. Okay, so I know that math went by fast. I encourage you to go through it more slowly on the slides later. Here's a picture. So this is the x-axis here is the effect estimate or the effect size. So the estimate is expected to be distributed according to this red curve, the null hypothesis, if there's no effect. And if there is an effect of size beta, it's going to be distributed according to this blue curve, H1. And now we're saying if you get a significant result, which means you have an effect size that's sufficiently large, so it's bigger than some threshold, and that's the shaded region here, how informative is that about there being a true effect versus not? Well, it's going to be the ratio of the area. The likelihood that you would get that under the null hypothesis is the area under the red curve, so that's this red shaded area. The likelihood of getting that significant result under the alternative hypothesis is the area under the blue curve, so that's the red, the blue plus the red area. And so the relative information, the information you get from a significant result about the alternative versus the null is the ratio of the areas, the blue plus the red divided by the red. So that's the rejection ratio. Here's a calculation of the rejection ratio for a typical psychology experiment. So this is using an estimate for a typical effect size in psychology from a big meta-analysis. And this is, the table is showing the number of people in the treatment group and control group, and for a significance threshold of 0.05, how much power you would have, and then what the rejection ratio would be. So it used to be taught in psychology as a rule of thumb that you needed 20 to 30 people per condition. If that was what you did, then for a typical effect size, you would have power between 40 and 50%. And finding a significant result would give you evidence of between 7 and 10 times, rejection ratio of 7 to 10 times evidence for an effect relative to null. If you want 80% power, you need 65 people per condition, and that gives you a rejection ratio of 16. That's 80% divided by 5%. So that means it's 16 to 1 evidence for an effect when you find a significant result. OK, so now applying this to genetic association with educational attainment, we need to choose a prior. And it's often when you're thinking about priors, useful to consider a reasonable range for priors. Well, here's where it gets a little weird that we're just having these two states of the world of an effect or no effect. It would be nice to have a distribution of effect sizes. But in order to try to give probability numbers to a prior for 0% null hypothesis versus the 0.02%, I'm going to take as starting points an estimate of the variance explained by the additive effects of the measured SNPs, a Gremel estimate. This is something that Patrick's going to talk about later. It's a kind of heritability estimate for the measured SNPs. And for educational attainment, we generally get 20% for that number, around 20%. And I'm going to use the fact that there's about 1 million independent SNPs in genome-wide data in European ancestry samples. So for one extreme of our prior, I'm going to assume that that every SNP that's associated with educational attainment has this effect size of 0.02% R squared. So since the total variance explained in educational attainment by these SNPs we know is 20% from this Gremel estimate, if each one has this effect size, then there have to be 1,000 of these SNPs. So if there's 1 million independent loci in the genome and there's 1,000 associated loci, then every locus has a randomly selected locus has a prior probability of 0.1% of being associated. So that's a lower bound, actually, on the prior probability of association, because most loci have smaller effects than this. This is the largest effect size. So there's more than 1,000 loci associated with educational attainment. Now, for the other extreme of the likelihood, for the other extreme for our prior, we're going to treat an effect size 10 times smaller than our 0.02% as essentially a null effect. And we're going to imagine that that's the effect size of all of the loci associated with educational attainment. So in that case, there are 10,000 loci that we have to divide this 20% over loci with that effect size. That gives us 10,000 loci. And that means that each locus in the genome has a prior probability of 1%. So there's a much bigger chance of any given locus being associated with a smaller effect size. OK, so equipped with that range of reasonable priors, and I'm also going to show in this calculation much larger priors of 5% and 10%, which may be what candidate gene researchers had in mind when they did their studies. And I'm going to do a calculation for 3 different sample sizes. So 100 is a sample size for a typical candidate gene study. 10,000 is what you would get studying, say, the health and retirement study. And then 100,000 was a GWAS meta-analysis, the sample size of EA1. The power associated with identifying an effect of size 0.02%, would be 0.05 for the sample size of 100, about 30% for 10,000, and almost 100% with 100,000. And what I'm showing in the table is, using Bayes' rule, how you would update to your posterior probability of there truly being an effect from each of the prior probabilities. So the first thing to notice is that in this first column, these posterior probabilities are essentially the same as the prior probabilities. That's because the power here is epsilon more than 5%. So the rejection ratio, which is this 5% plus epsilon divided by 0.05, is essentially 1. There's essentially no information from statistical significance when your power is that low. this extreme, we have power of 99%. We can infer much more. But the other thing I want to highlight is that even with a prior probability of 10%, we're only getting up to a posterior of about 70%. So there's still, at our 5% significance threshold, there's still about a 30% chance of a false positive, even with an essentially perfectly powered study. So why is that? Well, that is because, so first point I made is that when we have these tiny effect sizes, we're going to need large sample sizes to have adequate power. But this other point is about the significance threshold. It's another perspective for why we need a stringent significance threshold, a Bayesian perspective on it, besides the multiple hypothesis testing frequentist perspective for why we need a stringent significance threshold like genome-wide significance. And not only for GWAS, but even for a candidate gene study, we should have been using much more stringent significance threshold. So what's the idea here? Well, remember this formula, that how much we can learn depends on this ratio of power to significance threshold. And we are in a world of low priors. Like, 1% is an optimistic prior. The issue here is that power is bounded above by 100%. So even with our large sample, we can't do better than a power of 1, which means that at a significance threshold of 0.05, this rejection ratio is bounded above by 20. So at best, what we're going to learn by statistical significance is 20-to-1 evidence in favor of there being an effect. And in a world of low priors, like 0.01, 20-to-1 evidence just doesn't get you very far. So in a world of small priors, you need stringent significance thresholds in order for statistically significant results to be meaningful evidence against the null hypothesis. If we adopt the genome-wide significance threshold of 5 times 10 to the negative 8th, then we can do much better. Now power is bounded above by, now this ratio, this rejection ratio, power divided by significance threshold, is bounded above by 1 over 5 times 10 to the negative 8th, which means we can get 2 times 10 to the 7th strength of evidence in favor of an effect from significance. So that's big enough to overcome even the very small priors that we would have for genetic associations. So that helps explain why the GWAS results have been as replicable as they are. We can get credible findings from well-powered studies, even with pessimistic priors. So here's that same figure from before, now recalculated with genome-wide significance instead of 0.05. You have essentially 0 power even with 10,000 people to identify a true effect of magnitude 0.02%. And notice, even with 100,000 people, our power is not that high. It's 16%. But turns out that's OK. 16%, while we'd ordinarily think of that as low power, it's plenty when you have a significance threshold of 5 times 10 to the negative 8th for credible results, because 0.16 divided by 5 times 10 to the negative 8th is a pretty big number. So even with pessimistic priors, we can be virtually certain that we're finding an effect. Now, 16% power means we're only finding 1/8 of the truly associated SNPs with educational attainment when we have a sample of size 100,000. But the SNPs that we do find are very credible. Okay. So 16% power is perfectly adequate. Bottom line is that when we move into genetic association and gene discovery, we're moving into a world which is pretty different from the world we're used to in social science, where our priors tend to be reasonably high of finding effects, and effect sizes tend to be reasonably large relative to genetics, where the effect sizes are tiny and the priors are tiny. So our intuitions are off. And that's why, it's generally good to do power calculations and think about credibility calculations. But in the context of these genetic associations, it's especially important because our intuitions, we're just outside the normal range of where our intuitions operate. Okay, so these are important skills to use, and we're going to be drawing on them for the rest of the course. Okay, so sorry for running a little bit over. Yeah.
So just curious, what's your experience been with writing proposals for like federal funding, especially because I mean, if you identify as a social science genomics, has there ever been the situation where your proposals went to like a social scientist that maybe like was ignorant to the research and found that the R-squared value of 0.02 is just so little to find your research to be meaningful? So I'm just curious, what's your experience been like that?
Yeah, so the question is when you're writing grant proposals or giving talks to social science audiences, and you tell them I'm going to be studying effect sizes of 0.02%, might they find that problematic? Yes.
And how do you approach it?
And how do we approach it? So actually, when we published EA1, we viewed it as a big success because we were finding, we thought of ourselves as showing that the GWAS approach could work for behavioral phenotypes. We identified in that paper 3 SNPs associated with educational attainment, and they replicated well. And we showed that, so we thought of the paper as showing that we could overcome the replicability problems of the candidate gene studies. And we were estimating effect sizes that they were actually much smaller than we even expected before doing the study for educational attainment. They were very small, but they could then serve as benchmarks for doing power calculations for other GWAS. I think it's fair to say that the general scientific reaction to that paper was ridicule. I know of at least one case where a researcher showed our paper as an example of how ridiculous social science genetics is and GWAS, because the effect sizes are so small as to be meaningful. Like this paper is published in Science. It's like the effect size is essentially 0. So yeah, it's a challenge communicating with social science audiences and talking about this other world where these effect sizes are so different. I think you are fortunately in a very different world than we were then, because you can talk about polygenic indexes, which have quite a bit of predictive power relative to most variables that social scientists study. I mean, 15% R squared for the latest polygenic index for educational attainment is almost as high as some of the strongest sociodemographic predictors of educational attainment, and much, much larger than what's been found for interventions designed to improve educational performance.
So in this educational attainment study, there's in the front end there's some mention of how these genes, some of the SNPs are associated with like brain development and behavior. So there's like some sort of a basic science pathway that is somewhat alluded to. Is there like how much of a pressure is there to do that in order to justify that this means something?
So the question is, how much is it important to talk about biology, like brain pathways?
Yeah, and if I may add, like, so that was like the paper that was the GWAS paper, but also papers, like social science papers using those data. If you all have published eventually, have like how much of the data is there, like how much of pushback do you get to justify that, there is actually a biological pathway, it's not just like associations that we found.
So the question is, in social science, how much do you need to justify that, that there's a biological pathway? So my sense is that you don't have to do much in the social sciences. We're used to dealing with variables where we don't understand all of the mechanisms, and that's part of the, it's often part of the study itself is to investigate mechanisms for an effect. I think it is helpful to be able to point out that there are these GWAS that find, brain pathways and other, that there are biological pathways that are that seem to explain some of the effect. As far as publishing the GWAS, it used to be essential to have some biology in the papers, and that's because in the medical genetics world, GWAS and gene discovery in general was often viewed as primarily about learning biology, like finding drug targets, learning about how the disease system works. And so it's kind of a new idea that the main output of GWAS might be the polygenic index that you're going to use for other purposes. I mean, even in medical genetics, that's becoming better understood, that for clinical applications, having a predictive score could be useful. But I think, in EA4, we have essentially minimal biology, essentially no biology. And we make the argument that the results are important for other reasons. So I think going forward, it's going to be less crucial that be in there. But historically, it's been very important. OK, so let's take a 10-minute break, resume at 10 after the hour. Yeah, see you soon.